Good morning. Today is Monday, June 15th, 2020. My name is Ricardo Sandoval, and this is the results video for Lab 9, Environmental Influences on Microbial Growth, Salt Tolerance, and pH Testing at LAPU for the Summer 1 term for Biology 225 Microbiology. And here's just a quick little sign I made. I'm summarizing what I just said. So in this week's lab, there were two experiments. There was one that we talked about sodium chloride or salts concentrations in nutrient broth, and the other in which I had plain old nutrient broth that I adjusted to either an acidic or a basic or alkaline pH to see basically whether S. epidermidis and S. cerevisiae would thrive in high or low salts solutions, or an acidic, neutral, or a basic pH solution. So I'm going to start, of course, with the first portion of this where we're talking about sodium chloride solutions. So again, just to summarize what happened in that lab, I had a total of 6 test tubes. I had 2 pairs that were I believe, a 1% sodium chloride solution, a 7% sodium chloride solution, and then a 15% sodium chloride solution. And then, if I recall correctly, 4 drops of S. epidermidis and S. cerevisiae, respectively, were placed into each test tube, and then each test tube had to incubate for a minimum of 24 hours. In order for me to see if there was any growth or turbidity. In my case, after 24 hours, the broths were just as clear as they were on the day of inoculation. So my broths incubated for a total of 48 hours. And then here we are today. So one thing I did want to touch on that I also touched on in my experiment video is the concept of tonicity. Which has a lot to do with sodium chloride. How much or how little sodium chloride is in a solution. So we have hypertonic, isotonic, and hypotonic. So I'm going to talk first about hypotonic. And when we're talking about cells and the membrane, or not to confuse things, but we'll talk about this first. So we're talking about cells and how much pressure. So when we increase the amount of sodium chloride in a solution, it brings in more water into a cell, which causes the cell to expand, and it can expand until it explodes and dies. In an isotonic solution, we're fine. Nothing's going on. Everything's perfectly in balance. Versus a hypertonic solution, where there's a decreased amount of sodium chloride or salt. So water is being brought out of the cell that can cause, that can eventually cause, or just cause in general, the cell itself to shrink, collapse, and die. So in these 3 sodium chloride solutions, we're looking to see whether S. epidermidis and S. cerevisiae thrive in a hyper or hypotonic solution. A great example of an isotonic solution is 0.9% NaCl, 0.9% sodium chloride, which is also known as normal saline. If you are dehydrated, for example, and you go to the hospital or even in urgent care, they will give you an IV of 0.9% NaCl, which is normal saline, which is what's running through our bodies to hydrate. In wound care as a nurse, I use normal saline to irrigate wounds, to clean wounds, and it's also used to flush IVs. So 0.9% NaCl or normal saline is the ideal environment. It's the isotonic environment in which microbes can sometimes thrive. So I'm going to start with my S. epidermidis samples, and I'm going to start with the one at 1% NaCl. So 1% isn't very much here, so we can talk about perhaps this is a hypertonic solution. And as you can see here, there's a 1%. And this is clear. And as you can see in my experiment video, it looks pretty much the same thing as it did on my inoculation day. There's no turbidity here or anywhere in the tube. So I can conclude here that S. epidermidis does not thrive in a 1% sodium chloride solution, and it's negative for growth. Alright, so next up is going to be my 7% solution. So we're getting more into the hypotonic portions here. And so there's 7% S. epidermidis. And again, this is clear. I mean, you can see me through it. You can see anything through this, and there's no turbidity. Nothing here, nothing in the nutrient broth. So again, we are negative for growth here. So S. epidermidis does not thrive in a 7% sodium chloride solution. Lastly here for sodium chloride, for S. epidermidis, I have the 15 or 1.5% sodium chloride solution. And again, just like the other two S. epidermidis samples of the 1% and 7%, it's clear. No turbidity whatsoever, no growth, nada. So again, nothing here, nothing here, nothing here. So in a hypertonic or hypotonic situation or solution, S. epidermidis does not thrive. Of course, I couldn't test if it thrives in an isotonic solution. Perhaps it does, but because I did not have normal saline available to me. So now we're going to go on to the S. cerevisiae samples, and again, we're going to start with 1%. So there's your 1%, there's S. cerevisiae, and as you can see, it's relatively cloudy. There's some down here, some up here, and it's kind of foggy. I mean, not terribly, but it is harder to see through it. So again, this is a hypertonic solution because there's not much sodium chloride in here, so water can go out of itself. And in this case, S. cerevisiae does thrive in a 1% sodium chloride solution, so this is positive for growth. Now this is the 7% sodium chloride solution for S. cerevisiae, and relatively clear. I mean, if you compare my 1%, the 7%, so 1% is here, 7% is here, I mean, there is a marked difference. This is the 7% get in my hand, but there is nothing here, and it's clear. So at 7%, there is no growth. So this is negative for growth. S. cerevisiae does not thrive in a 7% sodium chloride solution. And then lastly is my 15, my 1.5% sodium chloride solution. So there's the 15, S. cerevisiae. And this is completely clear. After 48 hours, after 2 days of incubation, there's nothing. There's nothing down here, nothing here, and the nutrient broth itself is completely clear. So in a hypotonic solution, since we have an increased amount of sodium chloride, what could have happened, honestly, is that because we have more salts and the water is coming into the cell, the cells of the S. cerevisiae could have just filled up with water over the 2-day period, or even less than 24 hours, since I saw no growth after 24 hours, and they could have taken in so much water and then just died, which is why there's no growth. So that being said, so S. epidermidis at 1%, 7%, and 15%. So both at a hypo and hypertonic solution, no growth, no turbidity, nada. So perhaps it could be hypothesized that at 0.9% or even a different percentage of sodium chloride, it could grow, it could thrive. While with S. cerevisiae, it really did do well at a 1% solution. So at a hypertonic solution, it did not cause the cells of the S. cerevisiae to shrink or die, even though there was a lower amount of sodium chloride, which did cause or can cause some of the water from the cells to go out. And then at 7% and 15% or 1.5% of sodium chloride or salt, there was absolutely no growth, no turbidity for the sodium chloride. All right, so in the second part of this lab, there I tested samples of S. epidermidis and S. cerevisiae at different pH levels. And again, to summarize what I did here is I had 6 test tubes of just plain old nutrient broth that I adjusted the pH to both an acidic level, a pH of 5, and an alkaline or basic level, a pH of 9. I adjusted 4 of them. So for the ones that went to a pH of 5, the acidic test tubes, I inserted 1 drop of HCl or hydrochloric acid into them. And then for the ones that went to a pH of 9, 1 drop of NaOH or sodium hydroxide was inserted into those. For the 2 test tubes that remained at a pH of 7, which is neutral, nothing was added since those test tubes already were adjusted to a pH of 7. After I did adjust the pH, 4 drops, if I recall correctly, of the S. epidermidis and the S. cerevisiae went into the corresponding tubes, and they initially incubated for 24 hours. And again, I was looking for turbidity to see if there was some microbe growth. After 24 hours, I saw nothing. So the tubes themselves did incubate for a total of 48 hours. And here we are today. So again, the concept of pH is we have a pH scale. That on the left side, we have very acidic. On the right side, we have very alkaline. And in the center, we have things that are neutral. So again, this is my pH scale just in color. But here, we're not going to see colors. But as you can see here, well, if you can't see, it starts with a pH of 0 here at the red end, and it goes all the way up to a pH of 14. So things at a pH of 14 are very alkaline. Things at a pH of 0 are very acidic, and things in the center are very neutral. So, for example, let's touch on alkaline. I mean, one of the big nutrition crazes right now is alkaline water. That it's very hydrating, helps immune systems, and alkaline water is normally sold with a pH of 8.5 or higher. In the same respect, acidity, just as we talked about in the lab experiments, acidity or acids have been used for preservation, for example. So think of vinegar. That is quite acidic. I mean, if you taste vinegar yourself, it's very acidic, kind of makes you pucker. And what acidity does in a food preservation standpoint is it kills microbes that can't survive in an acidic environment, which then helps food preservation, prevents foodborne illnesses, prevents us from getting sick, and basically extends the life of food. So if you're a person who likes pickles, for example, pickles are preserved using vinegar and they last for quite some time. In the same respect, if you like pickled jalapenos or pickled carrots, same concept. So, moving on to the experiment. So again, the tubes themselves did incubate for a total of 48 hours. And then what we're looking for here is turbidity to see if the microbe can survive in an acidic, neutral, or an alkaline environment. So I'm going to start with the pH of 5 for my S. epidermidis, which, here we go. And as you can see, it's completely clear. There's nothing here, nothing here, nothing here. You can see through it. I can see through it. There's absolutely no growth whatsoever, no turbidity. So I can conclude here that the microbe S. epidermidis does not survive, cannot grow in an acidic environment. So here we have S. epidermidis at a pH of 7. And again, no growth whatsoever, which honestly I found quite surprising because there's no additives whatsoever, nothing I put in here, versus this tube that has a drop of hydrochloric acid in it. But there's nothing in here. And given that 7 is neutral, I mean, most people would assume that we would have something going on, but no, not a thing. After 48 hours, nothing here, nothing here, nothing here. So at a neutral pH, S. epidermidis does not grow. It does not thrive. And then here we have S. epidermidis at a pH of 9 on the alkaline portion of the pH scale. And again, nothing down here, nothing here, nothing here. So no growth, no turbidity, nothing happened whatsoever in any of the 3 pHs with S. epidermidis. So nothing on the acidic scale, on the acidic end of the scale, nothing at the neutral portion, and nothing at the acidic portion. So, I mean, it's quite surprising to see that nowhere does S. epidermidis fit. And this is something that grows on our skin. Normally, it's natural flora. So it'd be quite interesting to see what the pH of most people's skin is. All right, so moving on to S. cerevisiae here. So this is S. cerevisiae at a pH of 5. And I hope you can see, I'm going to adjust my lamp here to make things a bit easier, hopefully. So there's some turbidity down here. On my end, it kind of looks very dusty. And there's some turbidity here. I mean, you can't clearly see my finger, but you can. So it's somewhat cloudy. Not completely, not terribly cloudy, but we do have some growth, I would say. A very minor amount, but growth nonetheless after 48 hours. So it can be inferred or concluded that S. cerevisiae can survive in an acidic environment. So here's S. cerevisiae at a pH of 7. And interestingly enough, there's little dry bits throughout the test tube, which I'm not kind of sure what they are, but here we have some growth here, a little bit here. It's a little bit cloudy. So, some minor growth here at a pH of 7 at a neutral pH, which is quite nice, since that I would expect for a neutral pH. And then here we go, S. cerevisiae at a pH of 9. And similar to the pH of 7. So we have some growth here, some growth here, and then as you can kind of see as I turn the tube, some growth on the side. There are, some little solid bits that have dried. So I'm not 100% sure what happened here, but from a scientific perspective, we have growth on the acidic portion of the scale, the neutral portion of the pH scale, and the the alkaline portion of the pH scale for S. cerevisiae, while for S. epidermidis, it didn't grow in any of those pH conditions. In neither part of this lab did I experience any sort of difficulties. I was a little bit concerned when using the hydrochloric acid because of its corrosive nature, but from carrying out the lab, no difficulties in that respect. This concludes the experiment video for lab number 9. Thank you.